All right, everyone. Well, welcome to the hangout today with Jut Win. My name's David. I'll be your host for today. So Jut is a conservation biologist who has bushwhacked through the jungles of Belize and China, rappelled into the deepest volcanic pit on the Big Island of Hawaii, drove the world's driest desert in northern Chile, used cutting-edge instruments on board NASA aircraft, and traveled to the most remote inhabited place on Earth, which is Rapa Nui. This is all to learn more about caves and the secrets they contain. Jut has led over 50 research expeditions and served as the supporting personnel on nearly 100 expeditions around the world. So, without further ado, I'll pass it over to Jut.
All right, thank you very much, David. And what I'm going to do now is I'm going to launch, let's see here, the presentation. All right, great. Okay, y'all, so what we're going to be talking about here is, in celebration of Global Biodiversity Day, we're going to think about a few concepts that essentially drive biological diversity, and that is evolution, speciation, and those two things feed in directly into biological diversity. And because you have your very own personal cave biologist for the next 25 minutes, I'm going to be applying these concepts from a cave perspective. Let's see. Oh, shoot. Just got bumped out. All right. Technical difficulties. Here we go. Can y'all see that again?
Yeah.
Okay, good. All right. So first, let's think about evolution. So what is evolution? And this can be explained essentially with one word, and it's called a mutation. And mutations are the way in which genes in an organism change over time. And what happens is these changes are typically random changes. They're random changes in how the organisms how they present themselves from one generation to the next. And what happens when we observe these random mutations occurring in a given population of animals or plants, what happens is the environment is selecting that suite of genes of a given subset of that population that best fits the environment. And over time, what happens is that can lead to a speciation of it. That can lead to a different species. So we can look at evolution in terms of to put a finer point on this, we can look at it in terms of microevolution and macroevolution. And when we're looking at microevolution, we're looking at those small changes, those small tweaks in the genetic makeup of an organism over time. And typically what happens is those smaller changes are occurring to adapt to a given environment as the environment is selecting for that suite of genes that best fit that environment. And then when we look at macroevolution, those are your larger scale changes, and that's when an actual speciation event occurs, when a species evolves from another species. And we kind of see it on this linear continuum in terms of how evolution occurs, because that's called gradualism. When we see those small changes in genetic makeup over time, which can ultimately lead to a speciation event, which becomes, at that point, it becomes two different species. And when we think about species, I think the best way to understand what a species is to look at it from the perspective of the biological species concept. And this was put forward by this very brilliant ecologist who's also a bit of a philosopher, and he thought about this idea quite a bit, and he came up with this species concept. And the biological species concept is that it is basically successful interbreeding from one generation to the next. So what happens is you have two bats that let's say we have two pairs of bats, a mama bat and a daddy bat, and a mama bat and a daddy bat, and both of those pairs have pups, and then those pups can successfully interbreed, and then over time you have successful interbreeding from one generation to the next. And what can happen in evolution are these events called adaptive radiation. And this to me is one of the most spectacular parts of ecology. And what happens is when an organism arrives to a new environment, in this case, we're looking at Darwin's finches, when one species is believed to have come from mainland South America, traveled 300 miles across the open Pacific Ocean to land in the Galapagos Islands. And when that happened, there were a lot of available habitats for that finch to be able to occupy. And what happened is we observed this burst, this explosion in speciation from a single species to filling these various different habitats. And when that happened, you can see here, if you look at the beaks, and this is what Darwin was observing, is you saw those beaks changed over time. And what happened was that was indicative of the fact that we were seeing speciation occurring. And I want to just throw this out here because I spend a lot of time in Hawaii, I work in Hawaii, and the adaptive radiation event associated with Hawaiian honey creepers, I think, is dramatically underplayed unless you're actually in Hawaii and you know about this. Whereas when we were looking at the example here of Darwin's finches, 15 different species. In Hawaii, upwards of 40 different species in an adaptive radiation event radiating from that species that we see here identified as being species number 1. So now let's think about what determines species diversity. Why are there so many species in a given area, or why are there so few species in a given area? Now this is a picture that I took of Guangxi, China, and it's in southern China. You can see it's very tropical. And this is great because we can use this to discuss this idea that ecologists have thought about for many, many years now, called the latitudinal diversity gradient. And what that is just a fancy ecological term for saying that there are a lot more species around the equator than there are in the polar regions. And that's what ecologists have observed and naturalists have observed for hundreds of years now, is that you have a high diversity in the tropics, the tropical rainforests, and in the poles where you have more icy conditions, more Arctic conditions, tundra conditions, you have fewer species. And then there's also this more localized concept that we can apply to when we see diversity, high diversity or low diversity at a given site, in a given area. And that's called habitat heterogeneity. And once again, let's look at this image here of southern China. And you can see the mountains in the background. Now that's where I was working, up in the caves in those mountains. And you can see that they're covered in tropical forest. And you can see rock, and you can see that there's going to be different areas where there's going to receive more sun than others, given the aspect of the mountains. And then you look here in the foreground, and you look at, it's agriculture, and it's where people live. And you can see here that if we just think about it, in an area where there's rice paddies and orange orchards and a lot of people, there's probably not going to be as many species there as there's going to be up in those mountains in that pristine rainforest. So that is a great example of habitat heterogeneity. So let's look at these concepts, these concepts that drive biological diversity from a cave perspective. And you're going to get basically Cave Biology 101 right here, right now. Caves are zonal environments. And what you can see here is they're divided typically up into these 4 primary zones, from light to complete darkness along this zonal gradient going from left to right. In the light zone is where photosynthesis can occur. In the deep zone, it's the deepest part of the cave. It's typically defined as being, it's the complete absence of light. It often has a very stable temperature. There's little to no airflow. And another characteristic that is very indicative of a deep zone is it has a near water saturated atmosphere. So the back of the cave, if you have those deep zone conditions, you will see water droplets forming on the walls of the cave. Water is dripping from the ceiling. And if you exhale, You can actually see your breath because it condensates as you exhale. And that's a deep zone condition. And that's along this zonal gradient is how we see animals selecting habitat and how we see diversity unfolding within the cave. And here's some drivers of biological diversity. And oftentimes what we'll have is it will require a combination of these to result in higher diversity. And in some cases, when we lack one or more of these different drivers of diversity, we will have lower diversity in the cave. The first of which is water. And we know here on Earth, water is life. Water is the driver of life. And in caves, it is no different. When there are these wet conditions, that sets the stage for there to be higher biological diversity. This is another driver, photosynthesis. In the entrances of caves, as I mentioned earlier, you can have plants growing in the entrance. And what can occur when you have photosynthesis occurring within the entrances of the caves is you are essentially setting the stage for these relict habitats, these habitats that may be reflective of times long past. In this particular case in New Mexico, we're looking at an ancient ecosystem that dates back to the Pleistocene. And within it, we found insects that were believed to have come from that time frame as well. Another driver of biological diversity are nutrients, and specifically, and what we have here is we have these root masses that come through the cracks and crevices of the cave and of the ceiling of the cave and into the cave itself. And these roots can drive an entire ecosystem. We will see animals that are eating the roots, and those white things that you on the roots are fungi, and they're eating that. And then you'll have spiders and other predators that are eating those animals, and you have an entire ecosystem that unfolds just because of the presence of roots in a cave. Another driver are bats. And bats are a driver of diversity because they bring nutrients into the cave. Caves typically have lower nutrients, obviously, than what we would experience on the surface because there's no photosynthesis. And bats supplement the nutrients by coming into the cave. In this case, you can see here in very large numbers. They're not very polite. They don't go and they don't leave the cave to go to the bathroom. They have to go right there. And when they do, they form these large piles of what we call guano. It's bat poo. And this bat guano becomes a nutrient source for insects and can also fuel an entire ecosystem. This is an example of what guano looks like in a cave. And what we can also have, and what also supplements the nutrients within the cave and helps the cave ecosystem along, are when animals actually die in the cave. And when the bats fall from the ceiling for whatever reasons and die on the cave floor, that becomes another food source. So more food in the cave can result in higher biological diversity. So now that we know what the drivers are and we have an idea as to how the cave is broken down into these different zones in which animals actually will select habitat and can become highly or either high or low diversity depending on what those drivers are providing for the cave ecosystem. Now let's look at the three primary cave specificity groups. And what I mean by that is that these are three different groups of animals that are specified to the cave at varying degrees. So the first of which is called a troglazine. Now troglazines are animals that require the cave for at least a portion of their life cycle. Now in this particular case, we have bats and crickets, and these are perhaps the best examples. Bats will roost in the caves during the day. Crickets will den in the caves during the day, and they will both will exit at night to forage. But they have to have a cave or a cave-like environment for at least a portion of their life cycle. Our next cave specificity group are called troglophiles. Now, troglophiles are animals that can complete their entire life cycle underground, but they're not adapted to the cave environment. Now, these troglophiles can also be found in similar cave-like habitats, like underneath a log or in a rock pile. And what these troglophiles are doing is they're essentially trying to find those cave-like environments in which they can make a living in. And our final cave specificity group, and this is what got me into studying caves, are these animals. These are the coolest animals in the caves. These are troglobites. These are animals that are adapted to complete darkness. They live in the very deepest parts of the caves, as I mentioned earlier, where those cave deep zones where there's a near water saturated atmosphere. These troglobites often have highly reduced eyes to being completely eyeless. They're often depigmented. They'll have this white yellowish color. You can see here that they also have really long antennae and very long legs, and they're typically covered in hair. And those really long antennae and all that hair become their eyes underground. And when we think about those troglobites and how those animals ultimately became adapted to the cave environment, over the years, cave biologists have thought a lot about this, and we've put forward two hypotheses regarding cave restriction. The first one is called the climatic relict hypothesis. Now, this was applied to the more temperate parts of the world, not the rainforests, but the parts we would call temperate regions. Canada, most of the United States is a temperate zone. And basically what this idea put forward was that climatic change associated with the glacial, interglacial periods over millennia would drive species into caves. Now, what would happen was when those climatic shifts would occur, the animals would be seeking more favorable conditions underground. And over time, what would happen was the surface ancestors, their very closely related surface species, would go extinct, leaving that new species, if you will, in the caves, and over evolutionary time, it would become cave adapted. And then comes the second idea, is called the adaptive shift hypothesis. And this is something a good friend and colleague of mine, Dr. Frank Howarth from the Bishop Museum in Honolulu, put forward. He started observing these cave adapted animals in Hawaii. And he thought, where in the world did they come from and how would they evolve? The climatic relict hypothesis does not apply in the tropics. So what he hypothesized was that species were able to colonize caves because they were exploiting these unoccupied habitats that were underground. Because as the Hawaiian Islands were forming due to volcanoes, these lava tubes were forming because of volcanism. And when that happened, all these new habitats were open. And animals started moving in and setting up house. So what we have is over evolutionary time, we have this animal that is moved into the cave simply because it can. It's exploiting a habitat underground. And then it evolves into a cave adapted form. But you have a sister species on the surface that did not go extinct and is just happy as a clam doing its thing up on the surface. And one of the best examples, one of my favorite examples, are the wolf spiders of Hawaii. And you see here on the left, you have the cave-adapted species. It's called the small-eyed, big-eyed wolf spider. At right, we have the big-eyed wolf spider. The only difference between these two species are the eyes. The one that is restricted to the cave is slowly, over evolutionary time, losing its eyes. So now let's look at these concepts and these ideas that we just discussed by looking at cave millipedes in southern China and in the greater Grand Canyon region. So first, let's look at the caves from northern Guangxi province in southern China. This is where I was working. You can see Guangxi province is outlined in red in the southernmost extent of the country of China. You see Guilin in the northeast corner, and that's where we sampled 4 caves, and we did this in November of 2016. Now, what I'm going to share with you now are but a snapshot of the diversity of millipedes that we encountered in these caves. And that's because when you're taking pictures of very small animals, most of the time you don't get a lot of good pictures. And these were the best of probably 1,000 pictures that I took of millipedes. And you can see here that we're getting progressively cooler in terms of the forms and the way that these animals look as we're going from slide to slide through this subset of millipede species that we observed. This is a Glomerida millipede, that's the family name. And if you look at it, we're all familiar with what roly-polies look like. Those are actually called terrestrial isopods. And this looks like a roly-poly. And I thought it was at first, and then I flipped it over and I noticed that it wasn't no roly-poly, and it was indeed a millipede. Absolutely beautiful animal. Once again here, you can see it lacks pigmentation, it lacks eyes. This animal is cave adapted. And what I want to point out here in these next few slides are of these particular polydesmid millipedes. We're going to be looking at 3 different species of Aphanochodrus, and look at these little scoots at each segment along the body of the animal. You can see how they're flared, and they become progressively cooler as we go from slide to slide. And when you look at this, when an ecologist looks at this type of design on an animal, you think, why did this evolve evolutionarily? What was the driver that forced this animal to develop these scoots? And I thought maybe it uses its legs to grab onto the rock and pull itself close to it so a predator cannot remove it from the rock. But that's just a hypothesis. I have no idea what those flared scoots are for, but absolutely spectacular. And this is my favorite. And to me, this looks like an ancient Chinese warrior. And this is another really neat photograph of a millipede, completely cave adapted. You can see here the very long antenna, the long legs, no pigmentation, it lacks eyes. And those last 4 images that I just showed you are all new species. We're in the process of describing these new species. We have at least 5 new species of millipede. 5 of those were featured in this presentation. So now let's turn our attention to Grand Canyon. We sampled 17 caves. Now we should be thinking, I just mentioned that I only sampled 4 caves in China, but we sampled 17 in the greater Grand Canyon region. That's a huge difference in the number of different places that we visited when we compare China to northwestern Arizona. From 17 caves in northwestern Arizona, we found 1 cave-adapted millipede species. Only 1. So what's driving diversity? In Guangxi province, from those 4 caves, we believe we have as many as 18 different species of millipede. We only were able to show you just a few, once again, because of the quality of my photographs. We believe 10 of those are cave adapted, and 3 of those species were from the same genus. And the ones from the same genus, and we're looking at this picture here, you see the one that looks like the warrior, and then we have the other 2 below it, the ones with the flared scoots. Those are all of the same genus. They're all very closely related. We would call them sister species. From northwestern Arizona, once again, 1 species from 3 caves, but we sampled 17 different caves. So what in the heck is going on here? Is it habitat heterogeneity? Is it a latitudinal gradient? Is it nutrients? Is it water? Is it a combination of all those drivers of biological diversity that come together in this perfect storm in southern China, which permits so many millipedes to occur in those caves versus only 1 millipede species in Arizona? And these are the kind of questions that we think about as ecologists. Ecology is very messy. It's not like, okay, we can compare it to math, for example. Math, you have an equation, and you know what the correct answer is. And if you don't get the correct answer, then you know you have to go back and go back through the equation and recalculate everything to get that right number. Ecology, it's very messy. And there are a lot of different drivers and a lot of different things to consider when we're trying to figure out the drivers of biological diversity. I would hypothesize, these are my ideas, that it's probably those drivers of biological diversity that I mentioned earlier in the presentation. All of them are present to varying degrees in Guangxi province, China. And a lot of them are largely absent in northwestern Arizona. I can say, with where we found that millipede in northwestern Arizona, there were deep zone conditions. You could see your breath when you exhaled in the deepest parts of the cave. There was food, there was decomposing vegetation in the very back of the cave that was driven by flooding. And as a result, we were seeing those cave-adapted millipedes. Where those two conditions were not met in northwestern Arizona, we did not find cave-adapted millipedes. So there's a lot to think about. And I would say that what I'm showing you right here is in its very earliest stages of trying to puzzle everything together to be able to tell a more complete story, at least concerning this example, as to what drives biological diversity of millipedes in caves. And with that, I can take any questions that you all have.
Perfect. Thank you so much for that. So we'll go to the different classrooms now if you have for questions for Jet. In the interest of time, we'll just start with one question for each classroom. If we have enough time, we'll loop back around, but there's a lot of classrooms and we're running out of time. So let's start with Miss Shavaria's class. Can you hear me?
Oh, yes.
Perfect. So do you have a question for Jet?
What is the most interesting you found? Nice amount. What is the most interesting? Say it again so they can hear you. What is the most interesting you've seen?
I'm sorry, buddy, I can't hear you. Could you please repeat that?
What is the most interesting you've seen?
The most interesting thing he's seen?
Hmm?
What is the most interesting thing that I've seen? Wow. That's a good question. And you know, I would have to say it really depends from cave to cave, from region to region. For example, I was working in a cave in Belize, and we found footprints in the cave that we believe were jaguar. Jaguars use caves throughout Latin America. They're found quite commonly denning in caves. And for me, it was just really cool. But as you can see here with those pictures that I showed you, when there's a lot that I find interesting, and I pretty much find everything interesting to tell you the truth. But when you see an animal that's cave adapted like that, with the crazy elongated scoots that looks like a Chinese warrior, when you see an animal like that and you know that you're seeing it for the very first time, that is interesting. That is cool.
Awesome. All right, thanks. So we'll move on to Miss Canellis's class. Do you have a question for that?
Yeah, they do.
Got a lot of questions.
Yeah, they do.
That's awesome.
At first, they want to know if you're in a cave right now, though.
I'm in my cave. I'm in my house.
Krista, go ahead.
You see any scary things in the cave?
Have you seen any scary things in a cave?
No.
Like monsters? No, I haven't seen any scary. Well, you know, for me, I don't get scared that much. So yeah, in caves, not really too many things that would scare me. I would say when we're doing technical work, because a lot of the work that I do involves wearing a harness and being on rope and rappelling deep into caves. There are some pretty scary things that can happen. I've been rappelling into a cave, and the person on the surface knocked a big rock loose, and a big rock comes flying by me. Had that hit me, it would not have felt good. That's a little scary. That wakes you up if you're about to fall asleep. So yeah, I guess those were that would be about the only thing I could classify as scary. Getting hit by a really big rock, that would not be fun.
Okay, sounds good. And so we'll move on to Miss Kaleworth's class. Do you guys have a question for Jet?
Yeah. Yeah. Yep.
Hi, I'm Ella.
Hi, Ella.
Why are all of the insects and animals in there all white or yellow?
Okay, so the animals that we saw that had that whitish, yellowish color. Okay, think about it. If you are, okay, we've seen, let's think about centipedes, right? And centipedes are often have a lot of color to them, right? Some of them are very brightly colored. And typically what that's telling someone that might want to eat them is, don't mess with me, man, I'm poisonous. So that is sending a message to the other animals in the ecosystem. And if you're a bird, for example, and you're a male bird and you want to attract a mate, you have very colorful plumage to where you saying, hey, look how pretty I am. Yes, I should definitely have a girlfriend because I'm so pretty. That's what birds do. But if you're underground in complete darkness, those visual cues disappear. So you don't need to have color on your skin any longer. And that actually is costly because the animal has to manufacture that through as it grows. So what happens over time is it is able to shed that because it no longer needs to have color. It doesn't, if the millipede was once green, it no longer has to be green. So it doesn't have to, that's a cost. It's an energetic cost that it has to put into becoming green. So over evolutionary time, it's no longer needed, and it ultimately sheds its color and becomes what we call depigmented, or that yellowish or whitish color that you saw. Next.
How many different adapted species have you discovered?
Let's see here. Right now we have, well, cave adapted. Yeah, that's a different question. Okay, well, what I can say is I would actually have to really think about that. I can say from China right now we have 7 different new species which are all cave adapted. In northwestern Arizona, I think we have about 10.
Oh, we're on here 2 times.
Something weird happened. But in general, in terms of new species we've discovered in caves, we have 50 new species from northwestern Arizona. We have 10 new species from Easter Island, and now we have 7 new species from China. And some of those are cave adapted, some of those are not.
Are they all centipedes or are they all millipedes?
Oh, goodness gracious. Let's look at this real quick.
Okay.
Most of these that we're looking at here are all new species. That slide that I showed you first.
Oh, dang. And then my friend called that one centipede with the really long legs that you showed us, he calls it the spider worm.
I like that. Spider worm. I'm writing that down.
Oh, he's writing that down.
Cool.
Whoa. Are you ready?
Good job, Darren.
Are you I don't know. I think he's. Your beautifulness gets in there. Are you?
We have any more questions? David, we got folks on the far, or my far left, frantically waving hands.
Yes.
We
All right.
is the super boy. How many species of insects can be found in an average cave?
Oh, that's a good question.
That is a good question. Well, remember those different drivers of biological diversity that we were talking about? If there are none of those, if it's a dry cave with no nutrients, and there's something else that we didn't describe because I didn't have a good picture to explain it, temperature. If it's really cold and there's no water and there's no food, you might not find anything in the cave. And I've actually been in caves where we've studied the cave. We've looked everywhere for insects, and we did not find one because none of those drivers of biological diversity were there. But if you have food and it's warm and you have water, then you can have a lot. And in terms of how many, we would then have to think about where are we in the world? Where is that cave in the world? Is it in northwestern Arizona? Is it in China? Is it in Belize? Is it in, I'm going to Spain in 3 weeks, is it in Spain? And then we have to think about all those things to try to piece together. And that's where we start thinking about, environment. And what is the surface conditions like? And was it once? So there's so many things that we consider. As I mentioned earlier, ecology can be very messy. And those are the things that we have to figure out is what are those drivers of biological diversity? And how do those drivers result in having a lot of animals or a few animals? So it's not an easy question to answer. So I guess the easiest answer would be, I don't know.
So it could be anywhere from 0 to
A million.
A lot.
a lot. Okay.
Yeah.
Like a lot meaning hundreds of thousands?
Well, okay, so in Easter Island, I've identified caves that support up to 20, 30 species, different species. 15, 20 in caves in Arizona. And like I said earlier, sometimes none, sometimes one. But, and this is where we have to start thinking about how good are the bug hunters? Okay, I think I'm a pretty good bug hunter. But if I'm working with people that are new and they're brand new bug hunters, they might not be good bug hunters. And sometimes the bugs are smarter than we are. They can hide a lot better than we can. So they're really good at hiding, and we can't find them sometimes. So that's something else that we have to think about. So there's, like I said, ecology's a very challenging subject, but a lot of fun because, well, for me, I get to hunt bugs.
Yeah.
Okay.
Any more questions?
I think.
Okay. Do you have another question?
Have you ever tracked a species to watch it change every generation?
Oh, wow. That's a really good question. So we have this this new species of isopod, of roly-poly, that we found on Easter Island. And it was found, we looked on the surface. We hunted for bugs on the surface, and we did not find this species on the surface. We only found it in caves. And what was really interesting about this new species is that in addition to it only being found in caves, the only other sister species, closely related species to that species we found on Easter Island, were in Hawaii. And all of those sister species were cave adapted. So what was really cool about that was we were thinking, well, maybe if we lived long enough, we might be able to observe that animal in Easter Island caves becoming cave adapted. But in a lot of cases, those sort of changes are very difficult for humans to observe. We simply don't live long enough to make those observations. In some cases, those observations have been made. We have been able to see a new species evolve.
That's what we talked about.
That's been done on the Galapagos Islands with the finches. But in caves, haven't done it yet.
What made you want to become a biologist?
Oh, can I do it? I didn't get to say no. Please do it. Yay.
If they choose us.
Fun. Ask. Why did you want to be a conservation biologist? Oh.
Was the question, why did I become a conservation biologist?
Yes, that's what we asked.
I was, ever since I was you all's age, ever since I was a young boy, I was interested in animals and plants. I was that little boy that would disappear into the woods, into the ditches, into the marshes. I would flip over rocks and logs looking for animals. So I was always fascinated by the natural world. So for me, it's just, I never had to grow up.
Still keep going and studying, and now we have to.
Oh, gee.
What's the coolest cave you've been in?
The coolest cave that I've been in. I get that a lot, and I never have a good answer. I would say the coolest cave that I've been in recently. Remember the spider worm millipede that we saw?
Yeah.
Oh,
The cave that had those spider worm millipedes. They had probably hundreds of them. Everywhere I looked, I could see them, and they were just crawling around in the cave. And I was sitting there watching them. I just watched them for hours. I was just so fascinated. They were so cool.
Wait, so what are
I'm just wondering.
So that was really neat. That was one of the coolest experiences that I've had in a cave in quite some time. To watch the spider worm millipede crawl all over the caves, and just like I said, there were just a lot of them.
What's the largest?
Oh, he sees you.
Question.
Yes.
What was the largest creature you've encountered in a cave?
Let's see. I was in a cave in Arizona, and I came around a corner, and there were 4 pigs.
In a cave? In a
In a cave. Now these are native pigs to Arizona. They're called peccary or javelina. They can get pretty big. 70, 80 pounds, somewhere around there.
Boy.
And it was a very narrow passage that I was walking through. So I knew that I needed to get out of the way of them if they really wanted to leave fast, or I might be in trouble. So I just moved and got up against the wall, and they walked right by me. And actually, one brushed my leg as it was leaving.
Do you ever?
Here we go, Jordan.
Is it hard to breathe in the deep zone of the cave?
Could you repeat that, please? I'm getting a lot of other white noise associated with other classrooms, I think.
Is it hard
Hey,
to
Jack, can you hear me now?
into the cave?
Okay, yes, David, I can hear you, and I got most of your question just then. Is it hard to breathe in the cave?
Is it hard to breathe in the dark zone?
Okay, that's a really cool question, and I'm glad you asked it. Now, in some caves, in one particular cave called Bayliss Cave in Australia, it has bad air. So, and what I mean by bad air is it has high CO2. And what? We need oxygen to breathe, right? We don't breathe CO2. So if it's high CO2, it can be hard for humans to breathe. When my colleagues were working in that cave, I did not go to this cave in Australia, but when my colleagues went to that cave, that bad air, that part of the cave that had bad air, they could not spend a lot of time in that part of the cave. If they started to get lightheaded, they would go closer to the entrance until they could breathe easier again. So yeah, there can be bad air in caves, and that's just something that we have to be aware of. You know, I often describe working in caves, you have to have your spidey senses on the entire time you're in the cave. You have to just really be aware of everything going on. And if all of a sudden you get a little lightheaded, you have to think, okay, why am I lightheaded? And then you have to puzzle that out real fast. And if it's bad air, you need to move closer to the entrance of the cave where the air is better and seeing if that lightheadedness goes away. So yes, there can be bad air in caves, but I have not experienced it personally.
Yeah, Jet, sorry about that. I got completely ruled out of the call. I couldn't control anything or hear anything. So have they been able to ask you questions? I've been out for at least 8 minutes.
Yeah, they've asked a lot of great questions.
Okay, great. Yeah, no.
But for the most part, really good ones.
Okay, perfect. Yeah, so we're way over time, so I'll have to end it now. But thank you so much for staying so long and answering all of them. I appreciate it. Sorry about those difficulties.
Oh, not a problem, David. Not a problem at all. Or is it just you and I talking now, or we still have the whole group?
The whole group's here. I just was finally able to get back in and all right. Yeah, there you go. Awesome.
Right. Okay, so my parting message for the whole group is everybody, always keep your childlike curiosity. Never let it die. Always question everything. You question everything. That's what we do as scientists. We have to question everything. If someone tells us something, if they say the answer is A, we don't just go, okay, the answer is A. We say, why is the answer A? So question everything, y'all. And thank you so much.
Awesome. Great advice. And thanks so much again, Jet. Good luck with all of your research. I hope it continues to go well.
Excellent, David. Thank you. Thank you so much for your time, man, and for moderating this group.
Oh, yeah, absolutely. Thank you. All right. Take care, everyone. Make sure to check out our other Hangouts by Exploring by the Seat of Your Pants. And hope you enjoyed our National Biodiversity Day. All right. Take care, everyone.
Thanks, y'all.